Welcome to this next webcast in our series on antimicrobial peptides. With me today is Professor Zvi Hayouka from the Department of Biochemistry, Food Science and Nutrition at the Hebrew University of Jerusalem. Professor Hayouka, many thanks for taking part in this webcast.
Hello, thank you for inviting me.
Professor Hayouka, it is important for us to find better ways to keep food fresh and safe, while at the same time reducing the levels of processing. You have been seeking innovative ways to inhibit microbial growth in foods. Can you tell us about your approach to finding new antimicrobial agents for food preservation?
So I think this is a very important issue. And as we all aware that there are several estimations that in 2050, we will have a huge number of people on Earth, and it's going to be very challenging to provide food for all the humanity. So there is a huge interest in developing novel technologies, approaches to prevent food loss and actually improve food safety and extending shelf life. And here, my lab focus is getting into the field. And the idea of my lab is to develop chemical tools in order to improve the food safety and also to extend food shelf life. We have several approaches and several programs in my lab, but in order to achieve these goals, we first need to develop the active agents, the antimicrobial agents that will be used as food preservatives. We are also working on water treatment to prevent bacterial contamination in water treatment, because water is the basic of all the food chain from crops and also for processing. And also in collaboration with Professor Saul Boardman from the Hebrew University as well, we are working together in order to develop crop protection agents. So we will prevent food loss in the fields, and then we can use all the crops for consumption, and then we will have more food for the people and the humanities. And in order to develop this kind of agent, we needed to design it by taking several parameters or needs that are needed to address before the design of the new agent. First of all, the antimicrobial agent is supposed to be very cheap and easy to synthesize, because if we are talking about food or a crop protection agent, or even to design bioactive surfaces for bioactive packaging, or even just for food processing surfaces, you need a lot of the compound, and it has to be very cheap in order that it will be taken in consideration for the food industry. The second parameter was that it's supposed to be very active and broad active, also towards foodborne pathogen, also clinically isolated pathogen, and plant pathogenic bacteria as well. And the third one that I think it's the most important one, it's the resistance. We are really interested in designing a resistant-proof compound, so they won't be able to develop resistance. The bacteria won't be able to develop resistance. And in that way, we can make sure that our active agent would last for a long period of time without any resistance occurrence. So keeping that in mind, in my background, I'm a peptide chemist. So of course, I know a lot on peptide and antimicrobial peptides, that this is a very, very interesting topic. But I don't know if all of you knows, but on our skin or in many, many other organisms, the first interaction of the organism with microbes as fungi, bacteria, and virus is on the surface of the organism. And as part of the innate immune of each organism, there is antimicrobial peptide, there is a secretion of antimicrobial peptide, that these are the first shield to protect the organism. And this kind of compound that were discovered in the early 80s of the last century were a really, really interesting compound because we can find them in many, many organisms, plant, mushroom, dolphins, human, and many kind of organisms, and they all have several and different antimicrobial peptide with different sequences and different structure, but most of them interact with the bacterial membrane and disrupt it using several mechanisms, causing pores and destroy it, and in that way eliminating the pathogen. So when we observe that there are so many examples of antimicrobial peptide with different sequences and different structure, we wondered if the sequence is really needed, is crucial for the antimicrobial activity. What we did, we screened all the antimicrobial peptide and we observed that most of them contain cationic amino acid in order to interact electrostatic interaction with the bacterial membrane that is usually negatively charged. And they contain at least 50% of hydrophobic amino acid in order to interact with the lipid chain in the membrane in order to poke the holes and to disrupt the integrity of the membrane. So we assume that the most important amino acid in the sequence is the cationic and the hydrophobic amino acid. So what we designed the new approach to for developing or synthesizing antimicrobial peptide. That was done during my postdoc with Professor Sam Gelman from Madison, Wisconsin University in the US, and where we designed the random peptide mixture, random antimicrobial mixture. This is a combinatorial library of peptides that all of them contain the same two amino acid, one hydrophobic and the other one is cationic. And we generated this library by incorporating in each coupling step two amino acid. Usually when we synthesize peptide with a defined sequence, we will incorporate in each coupling step one amino acid in order to make sure that we are generating and synthesizing a specific sequence. But in our case, because we are talking about combinatorial library, we would like to screen all the option sequences that we can get. So we incorporated two amino acids, one hydrophobic and the other one is cationic. And by that, we can screen how the composition, how the stereochemistry, and how the chain length of the peptide affect the antimicrobial activity. And what we observed that when we used leucine lysine or phenylalanine lysine as a mixture in 1:1 molar ratio between them, with a chain length of 20 mer peptide, we generated a very active antimicrobial agent. We observed broad antimicrobial activity towards gram-negative and gram-positive, and also for clinically isolated, like antibiotic-resistant strain, and foodborne pathogen as Listeria and Salmonella, and many plant pathogenic bacteria. So that was really encouraging, and it was kind of surprising because most of the antimicrobial peptides that were discovered in nature or were isolated from nature, they were active or towards gram-negative or towards gram-positive. And our agent were active towards both a type of bacteria, gram-negative and positive, and it was very encouraging for us. So we have the synthesis itself by combinatorial library. It's very cheap and easy to synthesize because we don't need to purify, or we are not able to purify the compound end to end because it's a mixture of different peptide with the same chain length, but just the order of the two amino acid along the chain length of the peptide is different from one to another. So we cannot purify it, but we extensively characterize it by mass spectrometer and amino acid analysis to make sure that the synthesis went well. So they are really active and they are cheap and easy to synthesize. And now we are working on experimental evolution studies in my lab in order to estimate the resistance occurrence. And for that, maybe we will have another discussion later on next year to tell you more about it. But the idea is that we have a very effective antimicrobial agent and very cheap to synthesize. So now we can talk about incorporating it or immobilizing this agent on several surfaces in order to have a bioactive surface. And we actually did it with several kinds of surfaces as glass and polystyrene bead. And we generated the beads that were really active by eliminating bacteria in the presence of these beads. And actually, we built a column that we were able to purify or decontamination of microbial contamination in water by just through giving the water go through the column, and then we totally eliminate all the bacteria in water. And we actually established a new startup company that trying to work on the scalability and the regulation, but it looks very promising that it might be very efficient in order to provide healthier and non-contaminated water. In other projects in the lab, we also used our compound. We designed random peptide mixture in order to use them as a crop protection agents. By spray them after we evaluated the antimicrobial activity towards plant pathogenic bacteria, we developed several assays in the greenhouse, and we showed that in the case of Xanthomonas perforans, bacteria that cause a huge damage for tomato, that when we spray our compound a day before the inoculation, we could see a great reduction in the disease severity and the disease progression in the greenhouse. So this is in the direction of the crop protection. Another advantage of the antimicrobial peptide is that if we can treat them as a food preservative, they are going to be totally degraded or digested in our stomach, and then we can use them as a superfood that you can use the building block for new protein synthesis. So what we designed several random peptide mixture, and we incorporate them in food matrices as a turkey meatballs, for example, that the shelf life of this product is very, very limited. In the refrigerator, it's around 3 days. And we incorporated our mixtures in the meatballs, and we could see a great extension in the shelf life. What we did in addition to it, we wanted to learn what were the populations of the bacteria that were eliminated by our treatment. So we did a microbiome analysis for the meatballs with and without the treatment and along the spoilage. And it was very interesting to see what were the bacteria that were target. It also were foodborne pathogen and maybe several food spoilage bacteria. But this was the first example of the usage of random peptide mixture as a food preservative.
Professor Hayouka, can you maybe also speculate a bit about what innovative solutions for food preservation we could potentially find further in the future, or where do you see most promise for further research?
Yeah, that's a very good question, and we also we always think about it, but I think the usage of protein and peptide as a food preservative it's just the beginning, because it's very powerful tool that totally digested as long as you use natural amino acid during the synthesis. But what we are interested is exploring the synergism between our compound with a well-established food preservative. So for example, sodium nitrate is a very useful food preservative in the meat processed industry, but it's very carcinogenic and very harmful in several, of course, concentration. So the idea of our project is to explore if we could decrease the amount of the sodium nitrate in the system, and by incorporating a new agent as the antimicrobial peptide mixture. And we observed very interesting results that there is a synergistic effect between them. And if 1 + 1 give you 3 in terms of activity, so you might reduce the concentration of each compound, and especially the ones that is more harmful. And by that, you can reduce the concentration and the chances that it will have a harmful effect, but still preserve the activity of the powerful compound.
And at the moment, how concerned are you about bacterial resistance to antibiotics? Or do you think we've now somehow changed our approach in the past few years and we're heading in the right direction?
This is a tough question because the antibiotic resistance crisis is not over yet. And I think it's just the beginning, because you can see in the literature that there are more and more cases. They are still rare, but that patients come to the hospital and they infected with multi-drug resistance bacteria, and it's very challenging to treat them in the hospital because the resistance occurs. So it's not so common now, but unfortunately, I think that in the future it will become more and more. We will see more cases like that. So I think we should, all the community should invest more efforts, and funding agencies need to put more attention to this crisis and to develop a new agent in order to prevent the resistance occurrence, and especially to design agents that are resistance proof, or that we can see a benefit by using the new compound compared to the traditional antibiotic treatment.
We are fast now approaching the end of 2021. When it comes to your research, what are your goals or aspirations for next year?
Yeah, it's a good time to start and plan the next year, and to wish everyone happy new year and a successful year. But in our research, we are really interested in getting more to the development and to make more effort in the design of a new agent that would be resistance proof. So we have another project in the lab where we are working on bacterial cell-cell communication. This is a quorum sensing call. And by eliminating this system, it was shown in the literature that you prevent the bacteria from becoming a pathogenic. And by using this agent, you are not affecting the growth of the bacteria, but you just eliminate the ability of the bacteria to secrete a virulence factor. So imagine that you will have a bacteria that it's no more pathogenic, so we don't care that we have them in our system because they are not harmful. So I think it's very important to design new active agents as quorum quenching to prevent this cell-cell communication, but also to design new agents that would be more broad and efficient, and as I mentioned before, resistance proof. We are really interested also in studying our agent as a new antibiotic. It's not just food preservative or crop protection agent. In collaboration with Professor G from Illinois University, we are exploring together the potential of using the random antimicrobial peptide mixture for a systemic use as an antibiotic in mouse model for several human pathogenic bacteria. And we observed great reduction in the disease severity in pneumonia or bacteremia, which might cause a mortality rate in hospitals, also from Pseudomonas aeruginosa and methicillin-resistant Staph aureus, a very, very dangerous human pathogen. And we are really encouraged by the results, and we are aiming in expanding our research and learning more how our compound interact with the host, not just with the bacteria, but we are really interested in learning what our compound, how they affect the host immunity, and how we can use them in the future in a new direction.
Professor Hayouka, it was a great pleasure. Thank you so much for taking the time today, and let me wish you all the very best for next year and for your further research. Thank you.
Thank you very much. Thank you for inviting me.